You know, your immune system has some incredibly precise, unbelievably powerful weapons in its arsenal. Today, we're going to dive deep into one of the most elegant of them all, the classical complement pathway. This is the story of how your body spots an enemy, builds a molecular machine right on its surface, and then literally blows it apart. But really, think about it for a second. How does that actually work? How do you go from a single invading bacterium to assembling a microscopic drill that can punch a hole right through its defenses? Well, that's the central mystery we are going to solve right now. Okay, so the first thing you have to get is that this demolition crew doesn't just show up randomly looking for a fight. Nope. It needs a very specific signal to get started, a marker that basically screams, attack here. And that signal, it comes from another part of your immune system entirely. So let's meet the first responder on the scene, the C1 complex. The best way to think about it is as a microscopic scout. The biggest part, C1q, has these 6 sensor heads that are just constantly scanning for trouble. And what exactly are they looking for? Antibodies. Specifically, IgM or IgG antibodies that have already latched onto a pathogen, flagging it for destruction. This is just brilliant because it directly links your body's super specific antibody response to this incredibly powerful immediate demolition system. Now, not all of these antibody flags are created equal. An IgM antibody with its kind of star-like shape is ridiculously efficient. It offers multiple binding sites all at once. So just one single IgM molecule can grab on to C1q and kick the whole thing off. IgG is way more common, sure, but you need a bunch of them packed tightly together in the same spot to create a strong enough signal to get that C1 complex activated. Once that C1 complex gets a firm grip, it's like tipping over the first domino in a long, unstoppable chain. What happens next is this rapid-fire cascade of activation, one event triggering the next in a split second. And it's just a beautiful, precise sequence of events. When C1q binds, it physically changes its shape. This twisting motion forces its partner, C1r, to switch on. And as soon as C1r is active, it immediately turns to its partner, C1s, and activates it by snipping off a little piece. And just like that, you've gone from a passive scout to a fully active enzyme, C1s, that is primed and ready to get to work. So what's the job of this newly activated enzyme? Its mission is simple. Start building a powerful molecular machine right there on the surface of the enemy's cell. So here's what happens. The C1's enzyme finds a protein floating by called C4 and snips it into 2 pieces. The bigger piece, C4b, has this hidden superpower, a highly reactive chemical bond called a thioester bond. When C4 gets cut, this bond is exposed, and it acts as molecular superglue, letting C4b permanently lock itself right onto the pathogen's surface. Trust me, it is not going anywhere. Next up, the C1's enzyme does the exact same thing to a second protein, C2. The active fragment that's created, C2a, then finds the C4b that's already anchored to the surface, and the 2 pieces snap together. And this combination of C4b and C2a, well, it creates something totally new, something incredibly important. It's an enzyme called C3 convertase. We have now officially built the engine of the entire pathway. This machine's one and only job is to take that initial tiny signal and just amplify it into a massive, overwhelming, all-out alarm. And when I say amplification, I mean massive. A single C3 convertase engine can grab and cleave hundreds and hundreds of C3 molecules in just a few seconds. This is the moment a tiny whisper becomes a deafening roar. Okay, so with this engine now running at full throttle and hundreds of new C3b molecules being churned out every second, the pathway unleashes this devastating 2-pronged attack on the pathogen. First up is the direct assault. Some of those new C3b molecules actually attach back to the engine itself, which completely changes its job. It's now a C5 convertase. This new machine grabs a protein called C5, and the resulting C5b fragment becomes the foundation for the ultimate weapon. It recruits proteins C6, C7, and C8 to build a base. And then a whole ring of C9 proteins rushes in to form the drill itself, punching a hole right through the enemy's membrane. And this really shows you the 2 lethal outcomes perfectly. On the one hand, those hundreds of C3b molecules we just generated are now coating the pathogen. This is called opsonization, and it's basically like plastering giant eat me signs all over the bacteria for other immune cells to find. And on the other hand, you've got the membrane attack complex, or MAC for short. That's our microscopic drill. It forms a physical pore, causing all the cell's guts to leak out until the pathogen just bursts and dies. So yeah, tagged for disposal and drilled full of holes. Game over. Now, you're probably thinking, with a system this powerful and this destructive, what stops it from just attacking our own healthy cells? It's a great question, and the answer is the body has built-in safety brakes to prevent friendly fire. And there are multiple checkpoints, which is key. A protein called C1 inhibitor can shut the whole thing down right at the very beginning by just yanking the C1 complex apart. Then, our own cells are covered in proteins like CD55, which will quickly dismantle any C3 convertase engine that accidentally starts to form on their surface. And for the final line of defense, our cells have CD59, also known as protectin, which literally sits there and physically blocks the C9 drill bits from forming that deadly pore. It's a system with multiple redundant safety measures. So we've seen how this perfectly calibrated demolition crew works, and we've seen how it protects our own tissues. Its importance really becomes crystal clear when you start to think about what happens when it all goes wrong. This quote really just sums it all up perfectly. It's not some random attack, it's a precisely ordered enzymatic cascade. And most importantly, it's the critical link, the bridge that allows the memory and specificity of your adaptive immune system to unleash the raw, destructive power of your innate immune system. And that brings us to our final and maybe most critical question, because this system is a real double-edged sword. It is absolutely essential for fighting off infection, but it can be incredibly damaging if it's not controlled. So what happens when those safety brakes fail?